Ok, estoy muy feliz de presentar en Argentina sobre ALS y Tofersen. Yo hablo un poco de español, pero no puedo presentar en su hermosa lengua. Muchas gracias por la invitación. Okay, delighted to be here, switching, of course, to English. I'm Tim Miller at Washington University in St. Louis, and I'll present some updates on Tofersen and maybe a little bit of another study too. I always start with industry collaborations, in particular Ionis Pharmaceuticals, with whom I've had the pleasure of developing antisense oligonucleotides, and I'll talk about Tofersen, which is an Ionis developed antisense oligonucleotide, and also Biogen, with whom I've had the pleasure of running clinical studies, and I'll talk about a Biogen clinical trial. This comes from the Washington University ALS Center, where we focus on ALS care, clinical trials, basic research, and I'll talk to you about some of our work there. Many of you know the genetic landscape of ALS, and that most ALS is singleton or often termed sporadic, so one person in the family. But about 10% is familial. And among that 10% that's familial, 10 to 20% of that is SOD1 or superoxide dismutase 1. And so if you're doing the math, that's about 1 to 2% of ALS overall. In the United States, that's about 300 to 350 people with SOD1 ALS. So SOD1 is superoxide dismutase 1. It causes, as I mentioned, about 1 to 2% of all ALS. And it's a genetic change, one letter that's changed in your DNA. And I'm going to explain DNA, RNA, and protein in just a minute. It's typically not associated with dementia. And the SOD1 abnormal protein is toxic to the neurons. Tofersen is an antisense oligonucleotide that's designed to lower levels of SOD1 mRNA and therefore protein. And it is designed to treat ALS for those with SOD1 mutations. Okay, so I'm going to go through DNA, RNA, protein. DNA is like a book, a set of instructions for how to build and maintain a human body. You could think of this as a set of instructions similar to like how to build a house. The RNA is a rewritten set of instructions. So that would be, for example, the living room. And then the proteins would be the things you find in the living room, a chair, a rug, or perhaps a brick that's in the side wall. Okay, so the DNA, RNA, protein. When there's an abnormal gene, there's a change in that genetic code. The genetic code is a series of letters. And then there's a change in the RNA. And that change in the RNA leads to a buildup of toxic protein. Okay. So putting that all together, DNA is like a set of instructions rewritten into RNA, which is a set of specific instructions for how to make a brick, for example. But if there's an abnormal set of instructions, you might make an abnormal brick, as shown here. And then when that abnormal brick gets initially put into a house, for example, things are fine. But then after some years, there's some wear and tear that wouldn't be there otherwise. And eventually that leads to the destruction of the house, or in this case, destruction of motor neurons. So antisense drugs block disease by blocking the abnormal RNA. So they work at the level of the RNA, and they work like an eraser. We're going to remove that RNA that's leading to the protein that's toxic, and therefore that protein is going to be removed as well. This was studied in the VALOR trial. This is a trial for Tofersen with 72 people on drug and 36 on placebo. And there was a loading period, so 3 doses given in the first month, and then 1 dose given about once a month after that. Tofersen, like other antisense oligonucleotides, do not cross into the brain if they're delivered peripherally. So you can't give it in the blood and expect it's going to get in the brain spinal cord. It needs to be intrathecally delivered. And this is an image showing the spinal cord and the bones surrounding the spinal cord, your spinal column. The spinal cord ends about 2/3 of the way down the back, and then there's this fluid-filled space, which you can pull out fluid, but also introduce a drug, in this case, Tofersen, which then distributes all throughout the brain and spinal cord. And that's the way we're delivering this drug. One of the things we measured in this study was the ALS functional rating scale. Many of you will be familiar with this. It's a 48-point scale. You start with 48 points, lose points as you go. And there are 4 subdomains: bulbar, fine motor, gross motor, and breathing. And Tofersen slows ALS progression. Shown here is the group in blue that got placebo first, and then in red are the group that got on the drug first. And you can see that there's a slowing of the disease progression here. I'll comment that this dashed line is the open label extension. So this is the time when everybody went on drug. And we first looked at this time point out at 28 weeks, and we didn't see much of a difference. The differences become more clear out here at a year, for example, on the ALS functional rating scale. It also slows the loss of strength. Again, in blue are the group that started on placebo and at 28 weeks got on drug. In red, this is the group that started on the drug from the beginning and stayed on the drug. And we're looking here at handheld dynamometry. Handheld dynamometry is the summation of multiple muscle strength. And then we're looking at that over time. And what you can see in this red group is that there's stabilization. There appears to be a flattening of this curve, which would be unusual in the setting of ALS. And we looked into this more closely. And these are data now out at 2 years. We're looking at function and strength. So listed here, function, breathing, and strength. And there's the ITT. The ITT stands for intention to treat. That means everybody. And then there's a faster progressing group and a slower progressing group, and that's based on a biomarker called neurofilament, which I didn't explain today. But the group that's faster progressing has a higher neurofilament, slower progressing, lower neurofilament. And then we looked at the delayed start that was in blue. They were on placebo for the first 28 weeks, and early start with those that got the drug from the beginning. And I blanked this out because I wanted to remind myself to take time to think about, what do we expect to see? This is a group, we're asking the percent that has shown improvement or stabilization from week 0 out to 2 years. So who is stabilized? And what's really striking in these data is about 20% in the early start group have stabilized. So they got drug from the beginning. About 20% in these measures of people have stabilized or improved way out at 2 years. As many people will listening to this know, and unfortunately in ALS, the expectation at 2 years is not that things are stable, especially in 20%. And we ran the stats on this, trying to figure out how likely is it this is by chance. The answer is 0, with many, many zeros in terms of the statistics after that. This is completely unexpected that we would have this sort of stabilization and improvement in some people with ALS. I need to mention that there are side effects of this medication, as are listed here: myelitis, radiculitis, and others. These side effects occurred in 7% of people on the drug, not in the placebo. Okay, some conclusions. Lowering SOD1 lowers this biomarker neurofilament. I didn't discuss that today, but in my opinion, that represents substantial slowing of neurodegeneration. There's a 1.6-year increase in survival. I didn't show you that data today, but in the fast progressing group, that was very clear. And then there's unprecedented stabilization and in some improvement. Again, about 20%, 25%. The drug, as many of you know, has been approved in the United States. It's been approved in Europe, and Biogen is working hard at getting approvals in many different countries around the world. want to recognize the many people that have contributed to this study. I'm showing you here the many philanthropic and other organizations that have been involved. I want to give a special thank you and shout out to the study participants. These are the brave volunteers that signed up for this new drug. This drug was delivered about once a month. Many of them needed to travel long distances to come to see us or to come to other centers, since SOD1 ALS is relatively rare. So they would need to travel to come to the center. So I want to recognize both the participants and, of course, their caregivers and families. I want to also recognize the steering committee, in particular, Merit Sokovich, and all the principal investigators that are listed here, and my colleagues, Steph Fredette and Toby Ferguson at Biogen. One of the questions I'm often asked is, how does Tofersen relate to sporadic ALS? Many of you listening to this won't have SOD1 as part of your ALS. Like, why is this relevant to my disease? Why is this relevant to my ALS? And so I want to take some time to highlight and discuss that. First, the first most important point to me is that ALS is a treatable disorder. You, of course, need the right drug. For example, an SOD1 focused drug for SOD1 ALS. So we need the right drug, go upstream. But the fact that this form of ALS is treatable means that it's highly likely, in my opinion, that all forms of ALS are treatable. And now we as a community are re-energized to find that drug for many more people with ALS. And it's not just the academic community, of course, that's where I exist, but the pharmaceutical industries and others are looking at this saying, well, if you could treat SOD1 ALS, I bet you could treat other ALS. And so there's a lot of energy now in thinking about how to treat ALS and how to broaden the number of people that can have this really impressive slowing. Another important point is that biomarkers change with effective treatment. All the data suggested that they should, but this is the first drug to really show that that's true in ALS. So neurofilament is lowered. And that gives us an opportunity to focus on neurofilament in clinical trials. For example, at 6 months, you may or may not see a clinical change, but if you see neurofilament lowered, you know you're onto something. It might continue that study for longer and be able to see a signal at a much earlier time than we would be able to see otherwise. And there are some studies being run just focused on neurofilament. And we can debate the pros and cons of that, but having a study that really only reads out neurofilament and get a quick answer for multiple different drugs. So this is now known to be possible, and I think that we will see many more studies focused on this particular biomarker, but other biomarkers as well that have the potential to change in the setting of ALS. So this is, from a drug development standpoint, also fantastic news. This is a challenging concept, the idea that genetic therapies might be good for sporadic ALS. It's being considered for SOD1, being considered for FOS, all of these therapies that are these fancy new therapies for genetic disease. Might they apply to non-genetic disease? This is a really important research topic that we and others will begin to tackle. I should make it clear that when I think about this topic, I think about it as research. When I think about treating people in the clinic with Tofersen, to me, wearing my clinical hat, I'm treating people with SOD1 ALS. I'm talking about research studies that maybe could think about treating others. The other point is that ASO delivery to the CSF can be effective. We are learning how to use this type of therapy, antisense oligonucleotides. These are big therapies And delivery to the cerebrospinal fluid does distribute these throughout the brain spinal cord. And now that we know that it can work, you could launch multiple different kinds of ASO approaches. And these are already being used for sporadic ALS, not just genetic ALS. For example, the ataxin-2 trial, there's a trial focused on stathmin-2, UNC13. These are some things that are involved in the protein TDP-43, which I don't have time to talk about today. But my point is that the same approach is being used already for non-genetic forms of ALS, for all ALS. And then I'll just I'll end on a quick comment about how we identify new targets for sporadic ALS. This is one of the new things that we're thinking about here at Washington University in St. Louis. And I'm showing you the upper motor neuron, the lower motor neurons. These are nerve cells that go from the brain and down to the spinal cord, for example, the upper, and then lower, they go from the spinal cord out to the muscles or from the brain stem out to the muscles. And in ALS, one curious finding that's been recognized for years and years and years is that the eye movements are affected much, much later. If eye movements are affected early on in the disease, then we really would think about a different disease. Eye movements are affected, but 10 years, 15 years later. So can we use that information to figure out why these motor neurons, because these are motor neurons, they control your eyes. They look just about the same, have all the same machinery as the neurons that go, for example, to your arm. They control eyes, but not the arm. And so we're looking at this differential susceptibility of motor neurons by doing a deep dive into understanding those neurons that control eyeball movements, for example, compared with all the other motor neurons. And this is the type of study that we figure out these key differences, then we can develop a targeted therapy using those differences to turn neurons that affect movement of the arms and legs into neurons that may be protected like those that move the eyes. Okay, that's all I have today in terms of my update, and I'm more than happy to take some questions. Thank you for your attention.
Excellent. Thank you, Dr. Miller. Okay, so that is a lot of exciting information and a lot to digest as well. So let's start with approvals. I mean, we know that Tofersen has been approved in the US and in Europe, and people in the global south and the rest of the world are very anxious and really looking forward to have this drug that seems to be really effective for SOD1 at least. So do you have any, I don't know, it's not exactly your expertise, but do you have any information on potential approvals around the world for Tofersen?
I don't have that information. I would say that work is really led by Biogen, but I would just say that it's very active. Like, so we have heard from people. We do understand. When I look at these data, to me, it's very clear that this drug works, and that we need to be able to offer this to people around the world with SOD1 mutations. And so it's again, these discussions are ongoing, constant, and pushing. There are regulatory hurdles, and so one of the data that I just showed you on, for example, the changes at 2 years are not yet published, and those data that I've shown you today, once we can get that into print, which of course we're actively trying to do, I think that that'll help, because then you'll have kind of another formal document that shows the degree of change, the reversals. So I think that in my opinion will help, and we'll try to make sure that as soon as that's published, that we get that into the hands of people like you and others who are advocates.
Great, excellent. Yeah, we are really looking forward to it. And so we've been talking with other experts, and the topic of Tofersen always comes up, and they all say it is the first drug that not only slows down the disease for a while, but in some cases, it stops the disease. And in some other cases, it might even restore function. So this sounds like a miracle in ALS, but it is happening. We have Argentinians and people from other Spanish-speaking countries that are sharing their testimonies, their experiences with Tofersen, and uh, they are really experiencing how this drug is changing their lives by being able to do things that they were not able to do just some months ago. So this is really exciting. Do you think that this is going to lead to other victories in our research field?
I I do think it's going to lead to other victories. I mean, the stories that have come out about people treated with tofersen are really amazing. You know, the individuals that we have seen and people regaining function, people's improvement has been just really mind-blowing to see for someone who's been taking care of patients with ALS for the last 20 years. And we just don't see that. I mean, so it's fantastic. And of course, it's not everybody's getting, improvement, but I think people are slowing down. I think earlier is going to be better than later. We could talk about that, but a lot of people that we're treating clinically now have been treated early rather than late. And so we're seeing those successes in terms of our own center. I do think it's going to lead to other breakthroughs in ALS for the reasons that I mentioned. One, the academic and the pharmaceutical industries are energized. If you can treat this ALS, you can treat all ALS. So we just need to find the right drug. So that is, I I think, coming through many different groups that are trying to find drugs for ALS. Again, if we can do it for this form of ALS, I'm entirely confident we can do it for other forms of ALS. But we need to find the right drug. And, so part of that we need to find some of the root causes. We need to go as far, the way I think about it, as far upstream as possible. Like what's causing it and treat it at that level. So I'm super optimistic.
Following up on that, so first it has been so groundbreaking that we're not only talking about slowing down, stopping, restoring, we are also talking about even the possibility of preventing ALS SOD1. We have talked to Dr. Benatar, Michael Benatar,
Yeah.
a couple of
Yeah.
years ago. He's been and he still is running this pre-files trial with SOD1 carriers that are still asymptomatic. And so, what can you tell us about that?
Yeah, I mean, this is a really interesting trial. It's called the Atlas trial. It's an ongoing trial, and it's enrolling people who have SOD1 mutations who are asymptomatic, as you mentioned. And the idea is that they're being followed, and they're being followed both clinically and also with the biomarker neurofilament. And so if neurofilament goes up, this likely means that their ALS might be starting. Or if they become symptomatic, of course, then their ALS is starting. If they become symptomatic, they go right on tofersen. But if the biomarker changes, but they're still fine, they either get on placebo or tofersen. And the goal there is to see whether tofersen can prevent the occurrence of ALS. Now, I mean, it's a bit of a nuanced argument, a discussion about when does ALS start and what does that mean. But when we say prevent, we're talking about preventing the symptoms of ALS. And it's entirely possible that if you give tofersen at the very earliest stage, or just when there's a biomarker change, that you could prevent the occurrence of any weakness. If you use that as the kind of marker where most of us think of the beginning of the onset of the disease as the onset of weakness. And you might be able to prevent weakness altogether. So just a few sick motor neurons, you'd be fine. So that's the vision. Um, there are challenges with the trial just because it's tricky to do, but the vision is super clear and exciting. And I think we'll learn about this with SOD1 ALS, but this would be true for other genetic forms of ALS. And I think eventually, if you have the big idea, would be, well, how could we monitor people overall in general? What would be some signs that ALS might be developing somebody even without a genetic form of ALS, whether it's a biomarker or something else, such that we could screen for it and treat people at the very earliest stages?
Again, very interesting and very exciting. So last time we talked, I believe it was in 21, and so Tofersen wasn't approved yet by that time, and then there was a trial that was negative, and then finally we had this other larger trial that was luckily it was positive, which again tells us that if we wait enough, not only for Tofersen, but I think this applies for any other drug. If we wait enough, we can see some results, which is, an idea that is contrary to the usual urgency that we always have as patients or relatives. It's not easy to tell people to wait
Yeah.
with this disease, but examples like the one with Tofersen really is really telling us that sometimes we should wait longer. Don't you agree?
Um, yeah, I have a couple of comments on that. One, the primary endpoint for this trial that I presented was negative. It was at 28 weeks, and there really was a teeny difference or not much difference at all. Um, that made the approvals and other things more challenging. And of course, the approvals are focused on the neurofilament piece rather than that, clinical endpoint. And you're absolutely correct. When you look at a year or 2 years, or even on 3 years now where we have data, you see really striking differences, especially if you compare it with natural history. And you just see that we're, in my opinion, clearly slowing down ALS. So it does take time. It takes time to heal. It takes time for those motor neurons to rebound, perhaps a bit longer than we expected. I, and so I do think that we will have some trials where we need to run them longer. Having said that, we did see early on by 12 weeks, we saw neurofilament go down. So the biomarker had changed early on. And that's why we had confidence that we were on the right track. And so you could think about a study where you would use neurofilament as an early indication. Neurofilament's not down. Maybe we got to move on to the next thing. Neurofilament is lowered. Okay, hold tight. Let's see what happens clinically. So I think we're all beginning to learn how it is we're going to use the biomarkers and use the kind of read out those clinical data. And how do we look at some of those early signals and continue? There's been some interest in going back to some of the studies that were at 6 months called negative in terms of clinical endpoints. Oh, what happened to neurofilament in that trial? Maybe that's a trial that could have been continued longer, especially if there were a neurofilament readout. So I think um, again, the impact of this study on drug development is large because of all of those things that you have touched on in terms of length of the study and biomarkers change. But it's large in a good way, right? Meaning that we've learned a lot and now it's being incorporated into other studies.
Good. And you mentioned other studies that are also testing ASOs, the antisense technique, and there's also a FAST study that is going on that also appears to have a good potential. That is also working with antisense. I know that you're involved in some trials and in some other trials you're not involved. But what can you comment? What can you mention about all those different trials around the world that besides the first and are also working with antisense?
So there are many, many trials now that are using antisense oligos as a technique to manipulate RNA, which then has downstream protein changes. And um, first comment on the FUS trial. Of course, that trial is ongoing, so there's not a readout for that phase 3 trial yet. We'll say from the data that I've seen presented by Neil Schneider, who's leading that trial, Neil Schneider at Columbia University, um, it looks fantastic. So, just the smaller, individual cases that have been shown at conferences, look really good. So I'm also very optimistic about that study. Of course, we need the larger study and the broader readout. Um, but then there are many others that are going on. I mentioned STATHM2, UNC13, others that are happening. These are targets that are likely involved in all ALS, and I think we'll see. We don't have a readout. We don't know whether they've worked. Ataxin-2 was tried for sporadic ALS, and it looks like that was not effective. Now, there's lots of questions about why and maybe how come that didn't work, but um, it gives us opportunities to test ideas like ataxin-2 as a way to modify ALS. So there will be many more studies coming through using this type of approach is what I predict.
Finally, let's talk about the general landscape in ALS-MND research, in ALS-MND clinical trials. Uh, Besides Tofersen's victory, we also had a couple of setbacks lately in these last couple of years because of what happened to the AMX trial. In some way, what happened with the Darabon as well. After the symposium last year in Montreal, we came back with a pretty clear idea that it seems like the field is trying to really understand ALS. It's like we are taking a step back. I mean, we're still having our phase threes and our phase twos, but it's like the whole field needs to really take a little bit of a step back and try to really understand the different mechanisms of the disease and how this disease really works before attacking the goal again and trying to get those effective treatments. What can you comment on that? I don't know if that is your same feeling or not.
I think many of us have been working on the pathogenesis of ALS for a long time, and will continue to work on it. And I gave you an example at the end about our new idea about how to develop new targets for thinking about different kinds of motor neurons, for example, for something that's going to have a big effect. So I think it's ongoing. I mean, there's been a lot learned. There's some new things coming through that are absolutely fantastic. So I remain optimistic. I agree with the setbacks, and I agree that ALS drug development overall has been hard, and that's why I think the enthusiasm from the Tofersen story is going to carry over. I think people can see it now. People can see what will happen, and one of the breakthroughs in understanding ALS has been to understand this protein TDP-43, which is protein that's in the nucleus, it's in the center of the cell, and it becomes abnormal when in the setting of ALS. And it's probably true, 98% of people with ALS probably have some abnormality of TDP-43. And it does a lot of different things, this protein, but there are some signatures of the misfunction of this protein that people have begun to recognize. And they're true in models, but also true in people. I mean, that's the data that's come out in the last couple of years to show that we can see these signatures of downstream of this protein, that this is a problem. And that's where a trial like UNC13 and STATHM2 and others come come into play. So it's really the understanding of that fundamental uh, um, uh, part of the pathogenesis that has led to these other targets. So I think there are targets being generated right now, clinical trials going on right now that are based on really outstanding science. So I'm enthusiastic. I agree with you that yes, we've had some setbacks, and um, most of us are always taking a tough look at what's the science, what's out there, what do we need to do, and is one of the hats I wear is that I co-chair with Jeff Rothstein the Northeast ALS Consortium Scientific Advisory Board. So we meet 12 to 16 times a year with different companies and talking to them about their new ALS drugs and helping them think about that drug development. And there's I mean, of course, I can't discuss all the things that I see in those meetings in a public forum, but there's great stuff coming through, and smarter science, and then you know we're able to engage as the academic community to help guide some of those drugs as they come through. So I'm optimistic, though I think we have to be um, cautious in terms of our view right at this moment, in terms of because patients will ask, what's ready for me next month? What's ready for me right now? And I do think we need to acknowledge that I don't have something great right now, right? And that it takes time to develop new drugs. So, we have to be um, cautious about how we approach that enthusiasm for future, but also with the reality of what's available for people that, you know, this is Wednesday, people that I'm going to see tomorrow in clinic.
Very clear. Okay. So thank you so much, Dr. Miller, again for sharing some part of your very busy schedule with us. This is an outstanding contribution for our community. It's really helpful. It educates our folks in our countries, and so we are looking forward to hearing more news about your research and about Tofersen and all the rest of the research in which you are involved. So hopefully I might see you by the end of this year, either in Toronto or somewhere else. But we're looking forward to seeing you again. And thank you for keep pushing things forward for keep fighting for this cause. So thank you, Dr. Miller, for that.
Thank you. It's always a pleasure to talk to your group. And as I think I mentioned in the beginning, delighted to have something that will be in Spanish for people in the United States as well. So for people to be able to hear this discussion and see this, because presumably I'm now subtitled in Spanish, is really a service to that community in particular. So thank you. And again, pleasure to be here.
Same here. Thank you so much, Dr. Miller, and see you next time. Take care.
Okay. All right. Take care. Thanks. Bye-bye.
Bye bye. Thank you. Gracias.